All of you are making the time. Oh, we are recording. Great. So grateful that all of you could make it this evening. So I'm going to introduce our guest this evening. This conversation is going to be led by Dr. Judy Wu-Smart, who's coming from the University of Nebraska-Lincoln. Judy has degrees in zoology and entomology, and she's researched the effects of pesticide residue accumulation in brood comb on honeybee health. Her PhD research examined the effects of systemic neonicotinoid insecticides on honeybee and bumblebee queens and colony development. She's also taught workshops, including beekeeping and pesticide certification courses. She's currently an assistant professor and extension specialist at the University of Nebraska-Lincoln. In her role there, she is developing a pollinator health program to help understand the underlying stressors in bee health and their interactions with environmental toxicants. Her goal is to integrate her research and extension efforts with policy to inform the regulatory decision-making process by identifying risk mitigation opportunities and best management practices that will better protect beneficial pollinators in agricultural and urban landscapes. So with that, I can hand it over to Judy to talk about bee survival struggles.
Yeah, thank you. Thank you for the invitation. It's really exciting to talk to you guys about it's not like I was mentioning earlier, it's not going to be the most uplifting talk, but I think it's a really relevant and timely talk considering the bill that is going to be discussed in this next week. And a lot of the research that we've done and some of the work that we've uncovered is some of the stimulus for some of the changes in the recent regulatory landscape. So this is one of our apiaries. We are the state of corn and cows. And so I just want people to absorb something very pleasant before we go into a lot of kind of depressing news and data. So thank you. I didn't know if you guys were going to have time to pull together some of my background, but it's kind of interesting. I've been working on this topic of neonics since the early 2000s, working with pesticides and the development of these newer compounds and the differences that how they affect and impact bees. And so to really kind of summarize some of the research and the things that we've learned is that there's a lot of different ways that pesticides can affect bees. Most of the times we separate them by looking at individual effects, effects that occur on an individual bee or at the colony level. But there's also a number of discussions related to the direct effects or indirect effects. And so in this talk, I'm going to kind of cover all of these areas so you can better understand how chemicals in general, and particularly neonics, affect all of these areas in nuanced ways that can really affect the overall success of a colony. Now, neonics, this is what they're the short term for. These are neonicotinoid insecticides. There's a lot of different terms for them. They're chloronicotinics, they're nitroguanidine. So basically, to just kind of summarize what they really are synthetic mimics of the nicotine compound. And so like nicotine, they can bind to receptors, nerve cells within our body, which is a very similar structure as in insects. However, insects have very different subparts or subunits to the receptors that make it more selective. So that's one of the reasons why people like neonicotinoids is that it is much more toxic to insects than it is to mammals. So it's more safe for the handlers like humans and our pets and a lot of different other mammal systems, and has a very potent response for insects. So nice selectivity on that sense. However, where we're concerned is a lot of these chemicals can move and translocate to all parts of the plant, including where they're often expressed in the nectar and the pollen, and that's where our bees come exposed. There's a lot of different active ingredients within this class of insecticides. The most common ones that you might hear of are imidacloprid or cothianidin or thiamethoxam. But there are hundreds of trade names, meaning that there are formulated names that have this active ingredient plus, a bunch of other compounds in the formulation. So they will be sometimes referenced by their trade name and sometimes referenced by their active ingredient. When we're talking about seed treatments, these are the chemicals that are put on top of or surrounding a seed, and these are for crop production purposes. Oftentimes, there are multiple layers of chemicals on that seed coating. They're going to have general coating layers that help the compounds bind to the seed. And there's going to be a number of fungicides in addition to that insecticide. Now, these compounds are going to be systemic, meaning that they can move throughout the plant seedling, permeate all the tissues, and move into the nectar and the pollen. These neonicotinoids can also be sprayed via foliar spray or injected into a soil or injected directly into the tree or put into a water system, like your irrigation system, and fed to the crops in that manner. Because of that systemic action, these compounds can readily move into those plants. The problem is that they can also move off-site and into other non-target plants. And so there is a lot of concern surrounding the spread of these compounds and their persistence. So this is just a diagram to kind of show, again, a number of different methods of treatment for those compounds. But in addition to the permeation of those plants moving throughout the tissue, into the leaves, into the roots, and this is one of the reasons why they're so valued amongst farmers and gardeners that want to control root-dwelling plants or tissue-dwelling plants. They're harder to treat with foliar sprays. So these systemic compounds are better at getting those pests that eat the roots or live within the tissue itself. Unfortunately, these compounds also spread very easily in the environment, either through the air or through buildup through the soil or running into the water stream. And so there is a lot of environmental concerns related to these compounds that isn't as problematic for some of the older compounds. Some of them are much more short-lived, where these ones seem to have very variable persistence depending on the soil type and the conditions of water and etc. So in relation to these kind of vast a number of different ways that chemicals, these insecticides can affect bees, and generally the regulations mainly focus on the pesticide bee kill, the classic colony where you see a pile of dead bees like this picture shows. But what we're seeing with these neonicotinoids and some of these newer chemistries is that they're affecting the behavior, the development, the reproduction, or altering the habitat in ways that affect all of these other attributes that are really critical, right? From foraging to learning to communication, even development and reproduction and mating. These are social insects. So all of these are extremely critical aspects of the colony functions. So when we see exposures that don't actually kill the bees outright, these incidences or these exposure incidences are rather changing the behavior, development, reproduction, and habitat in ways that are still making it much harder for those colonies to succeed. So I'm going to focus on these examples of how these neonicotinoids can affect these areas. And then I'm going to kind of end with this really extreme acute lethal exposure example that has occurred in our state that's kind of launched all these new policies and interest in restricting neonics. So this video here is one of my observational studies in a shed. So these bees are free foraging. They can fly in and out through their tube. This is a control colony, a very small, what we call micro colony. She's queen right. And there's being fed a very small sugar syrup solution. This colony, you can see, is about the same size, but it's being fed about 50 parts per million imidacloprid. This is a very small sublethal dose and they're being fed every other day. So a general beekeeper who's opening up a colony might see that the bees are still alive, but they wouldn't notice this huge significant behavioral difference where those bees that have the imidacloprid exposure are not foraging, they're not caring for brood, and they're not really functioning as a normal hive. So what happens when a colony can't bring in food, can't care for babies, and can't lay eggs. So this is one that's a really great example how a pesticide doesn't have to outright kill the bee to harm a colony. And we're seeing a lot of this play out in a lot of these behavioral studies where the neonicotinoid compounds are not directly outright killing them. They are at high levels, but the more concerning thing is at low levels, they're having these significant behavioral impacts. So the video on the top right left is a queen with her abdomen sticking out, and her head is actually in a cell, and she's apparently fallen asleep. In contrast, the lower right video shows a queen that's actively looking for her cell, an open cell, so she can lay an egg. And so eventually you'll follow her, and you'll see she backs into a cell and lays an egg. And she has to do this about 1,000 times a day, right? They'll lay about 1,000 eggs a day. So what happens when a colony is behaving like the upper left-hand side colony? That queen who has fallen asleep or is cognitively impaired will not be laying at the same rate as this queen at the lower right. And that's one of the challenges is it is a numbers game. These are social insects with a number of complex colony functions. And so if there is no brood to replace the naturally aging foragers, then the colony will slowly spiral and dwindle over time. So it's not an immediate response or observation. It's almost like a failure to thrive because you don't have the newer generations to replace the naturally aging ones. So we know that these compounds can affect egg laying. This is just another example of how on the left-hand side, a normal queen will move far distances and lay a number of eggs, whereas the picture on the lower right is that queen where she kind of just moved in circles and fell asleep. And this is a 15-minute observation. So you can see a dramatic difference in activity. Now, one of the challenges with kind of studying chemicals and studying pesticides is that there is a huge distinct difference between laboratory assays, which are very distinct and controlled assays, to field exposures. And, you know, with the regulatory discussions, oftentimes there's arguments about whether or not we have field relevant exposures. And what does it even mean to be field relevant? Because it really depends on where the bees are becoming exposed. And I'll just explain with neonicotinoids, like many other chemicals, we can't think of it as being restrictively at risk in agricultural systems or safe in certain areas, right? To be honest, chemicals are being used in both urban landscapes and agricultural systems and open spaces quite heavily. And so unfortunately, that is really going to drive what type of exposure and harm those bees are experiencing. Where they're bringing back those contaminants are really going to influence that. And when we think about bees, the ones that are feeding on nectar are going to likely die out in the field, but the ones that are bringing back pollen are packing it on their legs, and they're able to bring that home. And so the difference of exposure then separates within a hive, because now who are the most likely individuals that are going to become exposed to contaminated pollen? Your nurse bees, right? And what do the nurse bees do? Well, they take care of the babies. So if you lose all your nurse bee cohort because of contaminated pollen coming in, that's going to affect the babies that they're supposed to rear. But as a beekeeper, you may not see any impact on that colony until 30 days later when you're expecting newer bees to replace those old foragers. And so it's very difficult to kind of diagnose when you have problems like that, or when you have persistent issues with these chronic exposures through their food stores or through their combs. So this is another project that we worked on where we literally took colony collapse disorder dead outs. So these are beekeepers that experience massive dead outs. We took their dirty combs, we tested it for pesticides, and we then cut them out and stuck them back into the comb and caged a queen to lay on both sides. And then we took the queen, excluded her to the bottom box, and then just monitored the development of the brood in both of those pieces of comb. So the only treatment is the chemical residues in the comb. And we monitored the development. Then we reared out those bees and we put them into different experiments. And interestingly, some of the combs had a lot of chemicals in it. So this profile that you'll see control box right there had the comb had a little bit of coumaphos, which is it's a miticide that beekeepers will apply. We don't know where it came from. We don't use it, but it's likely that it came in from a package or from transfer from other bees. To contrast, the test comb had 17 different residues in it, including 5 neonicotinoids. These are rather, they range from levels that would be deemed safe to levels that would be deemed harmful, right? The ones in red are compounds that are considered highly toxic. The ones that are orange are considered moderately toxic. However, not all of them are at levels that would be considered harmful. The problem is we don't know the mixture of having 17 different low-level compounds mixing together. But what we do see is that when we rear the bees out of this comb, this graph on the top, it says controlled larval development. By day 4, those eggs should all have developed and hatched into larvae, which it looked like about 84% did. And then by day 8, those small-scale larvae should be filling out that cell into, almost that pupal stage. And then day 12 and day 19 is when they're sealed cap brood. In this case, the test larvae that were reared in this dirty comb, this is what I call clean comb versus dirty comb. By day 4, only 20% of those eggs, 20% of those eggs remained unhatched. And then by day 8, almost 40% of those larvae were still in the stunted larval stage. And so I think I have a photo of that. So this is what we call delayed development in these larvae. Now, remember, the queen is excluded from this area. She's still in the hive, but she can't access these combs. So it's not a case where she went back and relaid and redeposited eggs, right? So we found that there were several other treatment combs, these dirty combs, that had similar effects. And some of the ones that didn't hatch, 60 to 80% of those eggs were just aborted. They were just eaten or removed. So this is a case where it's really going to cause and lead to spotty brood patterns. Spotty brood patterns decreases the efficiency of brood care for the workers, decreases egg laying efficiency for the queen, because now she's got to go all over the place to find available cells to lay eggs in. And it affects their ability to thermoregulate, because now you don't have a tight cluster of brood. It's scattered everywhere you could possibly lay eggs and rear brood in. So when those bees came out as adults, we noticed that as the combs were starting to be reused and reused, these chemicals were migrating from the treated comb, the dirty comb, into the less saturated control comb. Just like natural, meeting equilibrium, the areas of saturation, when you spray perfume on one end of the room, it slowly migrates to spread out where it can, right? And what we saw that in the dirty comb sitting next to the clean comb. And the effect of that was that the adults started to emerge later and later. You'll see that they normally emerge between day 20 and day 21. But the more we use those combs and the dirtier the control became, those bees started to actually start emerging at day 21 to day 22. This doesn't seem like a huge difference, 1 to 1.5 days difference, but it does have profound impacts on the colony, particularly with the mites. Now, if you think about this, this is, if you're not familiar with this type of graphic, this is the worker brood at its capped stage. So they just got into their pupal stage, and this is marking the number of days under a capping. And you can see the pupal development of the adult bee, and then she emerges out, the worker emerges out here. This graphic shows that at day 1, the mother mite goes into the cell, and she subsequently starts producing offspring. Her first offspring is a son. Then the second offspring is a daughter. Then the third is a second daughter and a third daughter. And so what happens is as the host is developing, the mites are also developing. And the first daughter mite is going to have to mature and mate with the male mite. And the male mite never leaves the cell, but the mature mated daughters will then have to leave the cell as the bee emerges. So when the bee emerges, that first daughter actually has a 94% chance of survival. The second daughter mite only has a 38%, and then the third daughter mite only has a 13%. And that's a kind of just because of the duration and development time of those workers. So it significantly lowers that chance of that mother mite reproducing a number of mated daughter mites to spread. Now, to think about how drone brood trapping works, you've got 3 extra developmental days, and this is our drone brood at the bottom. I don't know if you can see my mouse moving back and forth, but the drone brood is developing on this line, and this is just following the same pattern. But the difference here is with those 3 extra development days, that second daughter mite has a 94% chance of survival. The third daughter mite has an 84% chance, and so far and so far. So in drone brood, they can produce a lot more mites and cause a lot more problems. What I'm trying to make the point of is that this dirty comb in this experiment was actually causing these workers to develop a whole day to a day and a half late. So that is actually increasing the survivability of those second and third survival mites. So it can increase varroa, which is a problem. It can also decrease hygienic performance. One of the graphics I apparently forgot to include in here was that when these neonicotinoids are affecting those bees in the video, they're also inhibiting their hygienic performance. So their ability to sense mites in the cappings or disease cells and remove them so that they don't reproduce or those diseases become infectious. One other thing we did with these adults as they came out, we put them back in colonies. So these newly emerged adults were marked, tagged. They were reared from clean and dirty combs. And then we put them back into colonies that were manipulated. The colonies were either manipulated with a low varroa, normal nosema level, or a high varroa, high nosema level, just looking at worst case scenario. We put them both in those colonies, and then we sampled bees back at 2, 3, 4 weeks post-release, and we tested them for nosema. Just right off the bat, there were really no differences in varroa mite levels, but that's also because of the short duration of our study for this particular project. But what was interesting is that we tested each individual bee for their nosema levels, and then we were able to see how many of those 20 bees were infected with nosema and at what level. And what we found was it didn't really matter that one of the colonies was actually being inoculated with nosema. So we were adding 50 million spores in that colony. It didn't really change how those bees responded. You'll see in the orange, that's the control. There was about the same number of bees that were infected with nosema as there were in those colonies. So it wasn't likely the amount of spores in their environment, but more how vulnerable those individual bees were to the spore infection. Another way of looking at it was when we looked at the number of bees infected and the spore levels, we found that the dirty bees had higher levels of nosema at all weeks, and the infections occurred at younger ages, which is really kind of unique because nosema in a lot of the literature is really known to only exist in the older bees because that was the emphasis on where to sample for bees to test for nosema. And the most likely place that was suggested is to target the foragers because they're the most likely to get infected from these spores. But in this case, if the larvae are reared in dirty, pesticide-laden comb, they become more susceptible and at a younger age. And this aligns with a lot of other lab studies that show that, yes, if you feed them neonicotinoids, they do become more sick with nosema and viruses, causing immune suppression in these systems and allowing these other pathogens to really take over. So those were a couple examples of how these neonics can affect behavior as well as development and susceptibility to disease. I kind of want to go into reproduction and habitat and then talk a little bit about that worst case scenario that happened in our lab. So this landscape study, and this is a typical landscape. There's cropland and then there's sensitive habitat that we're worried about protecting. So this project was initially setting up habitat to assess whether tree barriers could mitigate transfer of chemicals from the cropland into sensitive forage as well as waterways. And I started my position in Nebraska in 2015. And every year after that, in this one honey producing location, I kept losing hives. It was just a very classic spill of dead bees out of these colonies. I tried to change the timing of when I placed the colonies in this landscape, but ultimately I would still end up with really heavy losses, 100% mortality. And so I spent the last 4 or 5 years following these incidents, trying to investigate what was the cause of these losses. And in the colony, you would see non-discriminate signs of crud, really spotty brood patterns, lots of jitteriness, trembling, inability to really function. And notice inside the brood cells, there's a lack of brood food. So, one of the regulators had asked, is it the pesticides causing the brood loss? And you really can't separate between whether it's a direct effect of the chemicals or the effect of losing the workers that take care of the babies and feed the brood. And so either way, there's some significant impacts on the babies. I was finding dead bees with their heads in the food cells, kind of mid-feeding, like they were dying as they were feeding. And so that was a strong indication that it was associated with their food stores that was causing these losses. So that's what led me to kind of start investigating what was going around in the landscape around those fields and around our area. We had a student collecting plants and testing them for pesticides as related to that project. And they were finding much higher levels, 3,000 to 5,000 parts per billion of this neonicotinoid in milkweeds closer to the waterways than the fields. And that was really peculiar for us because that was counter to our initial hypothesis. We even contested the results and had it retested at a different lab. It turns out it was right. A number of the plants that we were testing right along the waterways were showing up with extremely high levels of these neonicotinoid compounds. And that's what we started to meet with state and federal officials and found out that there's an ethanol plant 1.8 miles north of our honey house, which is labeled here. All those pink markers are where our apiaries were located, where we saw losses. And then this red box is where the ethanol plant was. All the green lines are where these continuous and intermittent ditches and waterways, which were, I guess, driving and moving the chemicals off this plant and through the yards. So unfortunately, when I met with our state officials and EPA officials, they suggested that it would be better if I had more hives on site. And I really questioned, how many dying colonies is needed for action, because I kept putting hives out and documenting their losses, and it just didn't seem to stimulate action. And what I found out was that they were really unable to assist in bee investigations because I had already previously ruled out farm pesticide applications. And the reason is that FIFRA, which is what the Fungicide, Insecticide, and Rodenticide Act, is a federal act where the pollinator protection regulations fall under. And they oversee the sales, use, and disposal of pesticide applications, but they don't deal with pesticide pollution. And so there was no resources or an ability for me to get any resources related to pesticide or pollinator protection because it sits under FIFRA, which does not oversee pollution problems. And so it was upon our lab to kind of really figure out where the bees were becoming exposed and having to test basically everything around there. And after our meeting, I put additional colonies out, 12 additional colonies all around those areas, and then proceeded to watch them die and record it and continually show them. So the pictures on the left are 1 week after initially putting and establishing these colonies on site, and the ones on the right are from our control sites. So this is when they told me about this ethanol plant was processing treated seed, and I knew nothing about it. So I started reaching out to all of our folks, including our NRDC folks that invited me to kind of engage with the beekeepers in this topic. So ethanol is usually processed and through fermentation, and there's liquid and solid waste that's produced. And in a normal production, it would be fed to livestock as distiller's grain. Well, in this case, they were receiving pesticide-treated seeds, and then they were allowing the liquid and the solid byproducts to be land applied as soil conditioners to farmers without their knowledge of the pesticides. And unfortunately, this was a huge problem because they were arguing that they don't create waste and don't need to contain hazardous waste because they provide an economically viable byproduct. So this is the facility, and I just want to show you that this is from the agrochemical companies, not from local farmers. Surplus treated seed that can't be sold on the market needs to be disposed of because they're no longer German viable for market sale. So this plant was using it as a primary carbohydrate source for ethanol production. This is what it looked like in terms of its scale. And they're receiving other crop seeds, but the majority is corn, as well as other pesticide-treated seeds. And you can see from the satellite imaging that the decolorant from the seeds was actually leaching into their lagoon, as well as their waste piles that they were creating outside. And so they were producing about 100 gallons of this pesticide-laden waste. And the levels were 100,000 parts per million. That video that I showed you affecting those bees' behavior was at 50 parts per billion. So this is the scale that we're talking about. And these are just images of them taking the liquid from the lagoons and land plying them as soil conditioners to nearby farms via hose, spraying it so quickly that it would just run into the ditches and move off property. So these are the wet cake piles that they created with no mitigation for runoff. And unfortunately, this is a really kind of disturbing story of systemic pesticide pollution, that they weren't really breaking any laws, unfortunately, because of the exemptions at the federal level. These pesticide-treated seeds are considered treated articles. So the regulations only occur when the chemicals are applied to the seed, and not after they leave the seed company. So when they entered the ethanol plant, they were not flagged as a pesticide, nor were they required to have any kind of pesticide containment or control like a normal chemical. Before this incident, nobody really had talked about how disposal occurs for pesticide-treated seeds. And just to show you, as part of our experiment, we had these sticky slides to look at this dust plume surrounding this area. When my student reevaluated and looked at the data according to proximity to the plant itself, we found that the dust blowing around a mile away from this plant was as high as 520 parts per billion, 10 times what those levels are affecting those bees. And then this is the wet cake that they were allowing farmers to apply, about 33,000 tons of it. And the levels up here were 550,000 parts per billion. So extremely high. The mixtures of these neonicotinoids and fungicides and the seed treatments are really unknown. And so this is kind of really frightening stuff in terms of its levels. Now, the chemical itself, the company itself was touting that they were a green recycling program. Interestingly, they self-touted that they were receiving 98% of all discard seed created by the seed industry in North America. So that includes Canada and Mexico. And they were all processing it for $0, green recycling disposal program. Now, the plant shut down in 2021 because of our advocacy efforts and this research that has really opened up the eyes of what it's not about the bees in this case, right? The bees are the canary in the coal mine, saying that this landscape is really, really messed up. And the residents living near there had also experienced a lot of physical ailments and problems. And so, with the problems with these chemicals, is that they are in these plants at different levels and persisting at different rates. And we really don't have a good understanding of how long and how problematic they are. But we do see them in these landscapes really messing up the insects. So this is me just walking around collecting milkweeds. Butterflies are not supposed to be that cognitively impaired, right? It's still alive, but it's totally messed up. So this is kind of what the challenges that we're facing. It's in the wildflowers. It's showing up in the pollen. Luckily, it's not making it back to the honey because, like I said, the bees will die out in the field. But what we do see is that it has lingering effects. This plant was shut down in 2021, and my students started to do a bunch of marked plan behavioral studies to look at changes in behavior. And they did notice that the green line is a clean location. The red line is the dirty location. They're both farms. But what you see is that these bees are not engaging in brood care. They're more associated with feeding. But more importantly, they're starting their foraging almost 10 days earlier than they should be. And so that is precocious foraging is an indication that all your foragers keep dying in the landscape. So now you need younger and younger bees to start taking over that role. So these are very nuanced but critical behaviors that are being impacted. In addition to the egg laying, I already showed you that one. What we did was we took queenless colonies. We made splits, queenless splits. We gave them the same eggs and the same bees. And the only thing we changed up was the combs from dead outs from this one location, from dead outs from a different location. And then we looked and see how they were able to rear their own queens, their queen rearing success. Obviously, the dirty comb colonies had a much lower number of queen cells produced and a lower number of queen cells that ended up mating and successfully laying worker brood. A number of them become drone layers or get lost in that mating process. So we did this in multiple locations with different nukes. It wasn't associated with the landscape. It was associated with those reusing of the combs, which we all do, right? We all reuse dead out comb to replenish new combs. But in this case, it was affecting queen rearing success, lowering it to 30% versus 82%. And that is pretty significant in terms of an impact from just the reusing of comb. So the burden of proof is too high for beekeepers, honestly. This whole incident has led me to realize that there's a big need for science policy. The systemic pesticide pollution issue, again, this is when chemicals move from the water or the soil into the leaves, into the nectar, into the plants. There are federal gaps on treated article that make it impossible for us to protect ourselves from this without local or state solutions. There's actually no regulatory division in EPA to address pollution in plants, despite robust regulations in air, soil, and water. We just don't have it. It's very fascinating. I'm going to skip through some of this because I want to go through the discussion part. But obviously, it's a huge mistake for them to have such a practice like this. And using the collaboration we've built from the One Health team, which is this concept where the health of the environment, humans, and wildlife are all interconnected. And so the research approach should look at these at a wide, holistic view. And the policy messaging has been really aligned well with this, right? Because not everybody cares about bees, but there are folks that are avid birders. There are folks that are really interested in conserving natural resources. The community members are concerned for their own health and the health of their animals and kids and family. And so everybody played a role in this. We had the researchers like myself identify it. But what's important is that we get media involved, as well as the public involved, as well as the policymakers involved. So once we started making sure people understood that this was happening, there's a lot of local coverage that stimulated the closure of the plant, even though they had been going through years and years of violation. It also stimulated the policymakers to immediately address these with those bills, these local solutions, to make sure it doesn't happen again. And so, with the social media, with the social journalism coverage, other avenues have started taking a reevaluating the use of these chemicals, and they're finding that now that they've been on the market for 20 years or so, they aren't really panning out to be as beneficial as they were marketed to be, right? They were marketed to reduce chemical use, to increase yields, and to be more safe. But what we're finding is that this broad prophylactic treatment is really not benefiting farmers whatsoever. It's not increasing yield or increasing or decreasing pest pressure like it was marketed for. So there has been this huge demand and need for science policy involvement. Since then, Minnesota, New York, Vermont, New Jersey, and even California have all put in what they're calling extended producer responsibility bills, meaning that if you create something that's harmful, you should be responsible for it from cradle to grave. So this disposal aspect has never been addressed. But if we make them accountable for disposal, they may very well rethink pre-treating everything and not giving farmers any choice whatsoever. And so those discussions are happening now. I think it's incredibly important for us when we talk about pesticide treated seeds. Often the conversation is distilled down to benefits versus harm. When we're talking to legislatures, this is also distilled down to profits versus harm, the profitability of farmers versus the harm to the ecosystem. And we're asking them to make really hard decisions, balancing the needs of their constituents with wildlife that may be voiceless or voteless. It's almost apples to oranges. And I really encourage people not to make that comparison, because the comparison we really need is need versus harm. Do the farmers need these products? And does that outweigh the harm it's causing the environment, their bees, and our ecosystem, right? And in that case, it isn't true. Farmers aren't given a choice, and it doesn't show that the insecticide portion of the seed treatments are really needed. And so what are we doing? We're not giving them a choice. We're not against farmers. We are for farmers' choice in making those decisions. And in Colorado, I did notice that there's a big a change in farmers wanting to go into organic farming or regenerative agriculture practices. And in those cases, if they want to transition to those operations, they don't have a lot of choice. So this is a way to restructure that conversation that doesn't pit us against farmers, but puts us on their side. All right. So I'm going to skip this because this is just a lot of information about all the different ways that these neonicotinoids can affect bees. But keep in mind, most of these are sublethal in its most detrimental problems. What I do want to say is that when we talk about chemicals and regulations, most of the regulations fall under chemicals that are cancer-inducing or not. And unfortunately, neonics are not cancer-inducing. So they seem to fall under this bucket of not so bad. But what we do know is in other systems, they can cause, like mammals, they can cause changes in weight, tremors, susceptibility to diseases, respiratory issues, cognitive issues, even stillbirths and reproductive issues, right? So a lot of times when I'm talking to legislators, I'm reminding them that cancer is not the only illness that I am concerned about. I don't want my kids to have asthma. I don't want to have reproductive issues. I want to be able to live healthy lives, right? And so these quality of life issues are not currently being addressed under our regulatory procedure now. And so when we talk about examples that I just shared, when we're getting exposure in the air, the water, the plants, or food, what is the impact of those chemicals at a chronic level? And what does it mean long term to our systems, right? We don't know. And so we need to be more cautious about the use of these chemicals, because even last year, there was a lot of these news articles showing that it's in our fruits and our vegetables. And there is just a lot of chemical residues in our own foods, which is not a good thing for us. So it's not always about the bees. It's also about what the bees are telling about the condition of our systems and our food. So these are just the last two messaging slides. Is we really need a united message here. Fighting amongst yourselves and potential allies makes absolutely no sense. So I see this a lot on social media. I see a lot of pitting against backyard beekeepers versus commercial beekeepers, which honestly, I understand that we have a very unique and diverse industry, right? Think about this. If we try to get master gardeners in a room full of corn and soy growers and ask them to come together with the same similar needs and asks, guaranteed there would be a lot of disagreement and a lot of separation, because people are going into beekeeping just like they're going into gardening and growing of crops for different reasons. But there is no other industry like ours where we have a mash of backyard beekeepers with real large scale migratory commercial operations that view their operations and their purposes in beekeeping very differently than our backyard beekeepers, right? There's no right or wrong reason for going into it, but there are way more things we have in common than we think. I also see a lot of disagreement between wild bees or native bees versus managed bees. But let's get real here, because the things that are going to fix the things that we can agree on is increasing forage and habitat and reducing exposure. So it really doesn't matter too much. I suggest that we need to stop fighting and nitpicking about the certain seed mixes or et cetera, but focus on the policies that are just going to bring more flowers to the landscape and less chemicals in that landscape. And that's the same with farmers, gardeners, and conservationists. It's not an either or situation. Because we want to conserve resources or protect bees does not mean we're anti-farming, does not mean we're anti-business, right? And then for beekeepers in particular, it's not just varroa and it's not just bad beekeepers. The poor varroa management does not explain widespread losses of birds, bats, and other bees, right? And a lot of times I hear that if someone complains about their losses, that it's their own fault. And that isn't always the truth there, because you can do everything right as a beekeeper and your bee losses may not improve. You may buy the most hygienic, what I call designer queens, but they won't reach their performance potential if their environment is not suitable. If they have a lack of nutrition or high chemical exposure, that's going to cognitively impair them, locomotively impair them, and stunt their ability to do anything in terms of pest and disease management. The policy changes are necessary to drive these changes in practice, encourage adoption of new ways. I know that's scary, but it is the truth. In order for us to incentivize that behavior, there needs to be some of these tools in the toolbox. What I hope beekeepers will come out of this is that there is a power in positive empowerment. We want to make people aware. We want to motivate people to do something about it. But we also want to build alliances and partnerships from a diverse group of people and interests, not just the bees, right? And so I'm hoping that looking at these problems from that One Health lens, we can really kind of spread the messaging that the bee losses are not the problems themselves. Yes, they are a problem, right? But beyond that, because not everyone's a beekeeper, they are a messenger, a harbinger of broader ecological stress that ultimately will affect our ability to produce food and live healthy lives. And so that's the messaging I hope people will bring to the table in these discussions. All right, that's what I have. It's a lot.
Thank you so much, Judy.